Swayam Prabha. Digital India. Educated India.
In the previous lecture, we have discussed the principle of isothermal titration calorimetry, which has become a method of choice by many physical chemists, biological chemists, pharmaceutical chemists and many others who are interested in measuring the heats of very small value. Today, we will further discuss how to extract thermodynamic parameters from an isothermal titration calorimetric profile and how to interpret those thermodynamic parameters. Let us take a look at this slide. We discussed last time that the raw data from an isothermal titration calorimetric instrument will be in the form of microcalorie per second versus time or microjoules per second versus time. This microcalorie per second or microjoules per second is in fact power. So, what this figure actually represents is the differential power which is supplied or reduced to or from the sample cell to maintain the temperature difference between the sample cell and the reference cell 0. So, let us take a look back again. We also discussed that the area under each peak will be a point over here, which is the amount of heat absorbed or liberated depending upon the reaction. And as you can map both these, slowly the amount of heat evolved or absorbed goes towards 0, and that is what is seen over here. And if we take this as a fitted data, now let me talk something about fitting of the experimental data by a suitable model. The experimental points in terms of the amount of heat liberated or amount of heat absorbed versus the injection number or versus the molar ratio does not recognize what is the mechanism of the reaction. What you are getting is the actual experimental data. Now, it is our smartness to come up with a suitable model which can fit these experimental data points and extract the thermodynamic parameters. Now, the commercially available softwares offer variety of models starting from single binding site model. What is a single binding site model? A protein may have binding site for a certain molecule, and it may have one binding site, it may have two binding site, or it is also possible that the two protein molecules may be shared by the same, the two binding sites or individual two protein molecules may be shared by the same ligand. So, therefore, different models are available which are single binding site model. That means, single binding site means 1 mole of the ligand binds 1 mole of the macromolecule, or it may be two binding site models in which one protein molecule may bind two ligand molecules, or there may be three binding site models or multiple binding site models. Then there are other binding site models possible For example, sequential binding site model. What is a sequential binding site model? Sequential binding site means there are multiple binding sites available on a protein molecule and the ligands will bind in a sequential manner. Means first binding, the ligand will bind to a particular binding site, then the ligand will go and bind to another binding site, then the ligand will go and bind to another binding site in a sequential manner. As I just mentioned, commercially available softwares are available for variety of binding sites models, and if those are not available, then one will have to come up with a suitable binding site model which can fit the experimental data points in a nice manner. Now, let us go back to the slide and look at this figure. The dark dots are experimental data points and the solid line is a best fit to the experimental data points. Now, as I just said that different binding site models are available, and let us assume that one binding site model has fitted nicely to the experimental data points. And then if I assume this solid line to be the best fit to the experimental data points, this difference corresponds to delta H. Of course, delta H depends upon the mechanism of the reaction and the slope reflects on the value of the binding constant or the magnitude of the binding constant. And the stoichiometry N, which is the number of binding sites on protein available for the incoming ligand molecule. So, after we have fit a suitable model to experimental, so after we have fit a suitable model to the experimental data points, we have the different thermodynamic parameters. And what are those different thermodynamic parameters? Let us take a look at the next slide. These derived thermodynamic parameters we will call as thermodynamic signature, that is delta G naught, delta H naught and minus T delta S naught. We will call these as thermodynamic signature accompanying the binding of a ligand with a macromolecule. And the beauty of the thermodynamic technique, which is isothermal titration calorimetry, is that all these three thermodynamic quantities, that is delta G naught, delta H naught and minus T delta S naught, or in other words delta S naught, can be derived from a single experiment. That is, we have to do only one ITC experiment. And what is that ITC experiments? Let us take a look at this figure. We may choose to add 25 injections of a suitable volume. The volume of injection can vary from 2 microliter to 10 microliter or more, depending upon how you optimize the experiment. So, that means in one single experiment, which may be constituting certain number of injections, we will get all these three thermodynamic quantities, delta G naught, delta H naught and delta S naught. In addition to that, we will also get the stoichiometry of binding. The stoichiometry of binding N, I want to say something about the stoichiometry of binding N, its determination. N is usually called as the stoichiometry of interaction, which is how many ligands, how many moles of ligand bind to how many moles of protein. And as I mentioned in the previous slide, it is possible to get the value of stoichiometry. The N value determined from an isothermal titration calorimetric profile will be equal to the stoichiometry of the interaction or stoichiometry of binding, provided all the concentration in the cell of the isothermal titration calorimeter is in its active form, or concentration is same as its activity. All the molecules which are present in the cell are in the active form. Otherwise, if the concentration and activity are different, then one may get a little different number, which really N may not exactly match the stoichiometry of interaction. So, this has to be kept in mind. Now, what we will get from an isothermal titration calorimetric profile is delta G naught, which one can calculate from the value of K, delta H naught and delta S naught, which will be in terms of some numbers with a certain magnitude and also the sign. The sign is very important, because it is the sign which helps us in identifying the nature of intermolecular interactions. How to identify those interactions? When protein, for example, let us see over here, when protein is interacting with a ligand, what we are getting is a complex of P with L. Along with this, we have delta H naught and we have delta S naught. Delta G naught has to be negative for binding to occur, because if delta G naught is positive, binding will not take place. Now, these delta H naught and delta S naught can be either negative or it can be positive. Both possibilities are there. And these sign helps us in identifying interactions. Let us take a look at this figure. The red color bar represents delta G naught, the green represents delta H naught and the blue represents delta S naught, but in the form of minus T delta S. So, that means this delta H and delta S in the form of minus T delta S are the two contributions to delta G. The downward is represented as negative value and the upwards bars will be representing positive values. Let us look at the first panel. When delta G naught is negative, it has to be negative for binding to occur. And if delta H naught is also negative and delta S naught is positive, remember that we are talking about minus T delta S naught. If minus T delta S naught is negative, means delta S naught is positive. That means if delta H and delta S are both positive, it is predominantly indicating occurrence of hydrogen bonding interactions. It is possible that side by side there may be hydrophobic interactions, but a negative value of delta H is suggestive of polar interactions, which could be predominantly hydrogen bonding interactions. And if there is a predominance of hydrophobic interactions, let us take a look at the middle panel. Predominance of hydrophobic interactions are associated with endothermic effects. Delta H is positive and whereas delta S is also largely positive. Remember that hydrophobic effects, when we talk about hydrophobic moieties, this can largely affect the structure of water, because hydrophobic groups would tend to be away from water structure. Therefore, wherever there is an occurrence of more hydrophobic interactions, the entropy changes will also be significant. Now, let us take a look at the last panel, where delta G is negative, delta H is negative, but delta S is also negative. Minus T delta S is positive means delta S is negative. These trends are representative of conformational changes. Conformational changes can be in the ligand also, and conformational changes are also possible in the protein. We will discuss about these conformational changes a bit later, and also we will also discuss how to connect these thermodynamic quantities with the conformational changes a bit later. This is about how to extract the thermodynamic parameters from an isothermal titration calorimetric profile, and then what kind of information these thermodynamic quantities can give it to us. What we have started now appreciating is that the thermodynamic parameters which we derive from ITC are not only just telling us about the extent of interaction. Extent of interaction is reflected in the value of binding constant. Binding constant is equilibrium constant. And if the binding constant is high, means delta G naught is largely negative. So, we are not only trying to connect with the extent of interaction, we have also started connecting the signs of these thermodynamic quantities with the nature of intermolecular interactions. So, slowly and slowly we have started unraveling the power of isothermal titration calorimetry in giving the insights of the intermolecular interactions. Before designing an ITC experiment, an optimization of the experimental conditions is important. By now, large amount of data on variety of molecules is available where isothermal titration calorimetry has been used. And based on that set of experimental data, a general equation has been suggested which should help us in designing an isothermal titration calorimetric experiment suitably. Let us take a look at that. Literature suggests that if the affinity increases, that is, if we move from weak binders towards strong binders, the concentration requirement of the macromolecule decreases. That means, if the binding is weak, we will require large amount of the macromolecule, and if the binding is strong, we will require small amount of the macromolecule. That is, weak binders, if I say the binding constant of the order of 10 raised to the power 2 or 10 raised to the power 3, and strong binders means binding is 10 raised to the power 8, 10 raised to the power 9. So, if the binding is weak, we will require millimolar quantities of the protein, intermediate micromolar quantities of the protein, and very strong binders we may require only nanomolar quantities of the protein. And the general expression which helps in designing the isothermal titration calorimetric experiment is given in this slide, which is a factor C. C is equal to the stoichiometry of the reaction, N, which is the stoichiometry of binding, into binding constant, or I can also call it as association constant, into the total macromolecule concentration. And macromolecule usually is protein in this case. So, C is N times K times the total protein concentration or total macromolecule concentration. And the figure shows how the output or isothermal titration calorimetric profile will look like for different values of C. And this figure represents a range of C value from 0.01 to 1000. This one, let us take a look at this line. This is corresponding to a value of C which is equal to 0.01. This is for 0.1, and for a C value of 1, yellow line is observed. For a C value of 10, the green curve. For a C value of 100, the bluish curve, and for a C value of 1000, this violet type of curve is observed. So, what is the main observation here? The main observation is that when the C value increases, the shape of the isothermal titration calorimetric profile also increases. When C value is 0.01, you see it is just like a straight line, and no thermodynamic information can be derived from such an experimental data. Even when the C value is 0.1, it is also like a straight line and it does not give any information. For a C value of 1, some small curvature has started appearing, but still this is not sufficient to give an accurate estimation of all the thermodynamic quantities that we would like to derive from an ITC profile. When C is equal to 10, a reasonably good isothermal titration calorimetric profile can be obtained, where you have a good amount of data in the pre-saturation region and towards the saturation and intermediate, where the binding sites are getting filled. For a C value of 100, which is bluish line, this is also a very good isothermal titration calorimetric profile. But when C value becomes 1000, here we see a large amount of cooperativity kind of profile is seen, but a C value of 1000 has to be avoided for the reasons that I will describe a bit later. But a general conclusion that can be drawn from this kind of figure is that C value, a proper C value has to be maintained if we want to get a good experimental output. Let us take a look at the slide now. What is the best range recommended in terms of concentration? Now, here you see I have defined C as total protein concentration divided by the dissociation constant. And as I earlier mentioned, physical chemists would like to talk in terms of association constant, and biochemists would like to talk in terms of dissociation constant. So, therefore, we should not get confused when we are talking about association constant or when we are talking about dissociation constant. A C value between 10 to 100 is usually considered very good and gives a very good isothermal titration calorimetric output profile. A C value between 5 to 500 is okay, is good, can fall into the category of good. A C value between 1 to 5 and 500 to 1000 is okay type, not good, not very good, but okay. And a C value less than 1 and greater than 1000 we do not want. So, therefore, if the C value can be maintained between 10 to 100, that means in that case, under those experimental conditions, the isothermal titration calorimetric profile will turn out to be very good. And let us try to appreciate from this equation that increasing the value of C means we will have to increase the concentration of the protein. And also, as I mentioned earlier, that many times we may not be able to afford a large amount of protein. Now, this range of C values for optimizing the isothermal titration calorimetric profile is based upon large number of experiments. However, several reports are appearing or have appeared in literature where different authors have said that it may be possible to obtain good quality thermodynamic data even if the value of C is low. So, therefore, we must remain in touch with the literature for any updates, that is, if there are new experiments being designed or if there is a new information on how to get good thermodynamic data when the C value is small. So, in the design of an ITC experiment, a high C value will give an output like this. If C value is more than 1000, we have experimental data points in the beginning and we have experimental data points when all the binding sites are occupied, and in between we have no experimental data point. That means all the information which is occurring in this range where the binding is taking place, actually taking place or saturating the binding sites, that information is lost. Therefore, this kind of experiment is no good to extract the value of binding constant. This kind of experiment is only good to give accurate value of delta H, because delta H is difference between this and this. That is accurately known in this kind of experiments, but the information on K is completely lost. You cannot draw the value of binding constant. C value between 10 and 100. You see, we have good number of experimental data points and the model also fits nicely. So, therefore, these experimental conditions are preferred. This is a good C value. And if C value is low, then we usually will not have the initial data points. That is, what happens is, if you look at this, the binding constant is so large that as soon as you add small amount of ligand, it is completely binding. And the next injection also, whatever ligand is added, that is also completely binding, which is not happening here. When you are adding some amount of ligand, it is partly binding and partly going into the solution, because the value of K is not very large. So, for a weaker binding affinity, usually this kind of isothermal titration calorimetric profile is observed. The effect of C value on shape of ITC profile can also be seen in this figure. The upper panel shows the main isothermal titration calorimetric profile, and these are the dilution effects. And you see, if the value of C is increased from 5 to 40, the shape of the isothermal titration calorimetric profile also increases. So, we can clearly see the effect of increasing the value of C. But increasing the value of C comes at a cost. And what is that cost? That cost is the concentration of protein. The concentration of protein has to be increased. So, therefore, nowadays there are efforts by the different companies who manufacture the isothermal titration calorimeters to reduce the volume of the sample cell. But let us remember, by reducing the volume of the sample cell means we are also compromising with the amount of heat that is either liberated or absorbed. But nevertheless, if the sensitivity improves, this factor can also be compensated. So, what are the different isothermal titration calorimeters available? VP ITC with a cell volume of 1.4 ml, ITC 200 with a cell volume of 200 microliter, peak ITC which is an automated isothermal titration calorimeter with a volume of 200 microliter, nano ITC with a volume of 1 ml or 190 microliter, both are available, and an automated affinity ITC again with a volume of 1 ml or 190 microliter are available. So, what we discussed in this lecture is how to extract the different thermodynamic parameters from a single isothermal titration calorimetric profile, and that it is not just the number, the signs are very important to connect these thermodynamic data with the nature of intermolecular interactions. We also discussed how to optimize the experimental conditions so that we get a very good isothermal titration calorimetric profile. We will discuss several applications of the isothermal titration calorimetric data in the lectures ahead. Thank you very much.